In fact, there exists a highly efficient motor with energy conversion efficiency of virtually 100%. What is more, it rotates at a maximum speed of 1,500 revolutions per second, far faster than even the fastest Formula One race car engine. A bacterial flagellar motor with a diameter of only 40 nanometers. It is composed of protein molecules. 2 micrometer-sized bacteria rotate its flagella of 10 to 15 micrometers in length to achieve motility.
Now, in terms of the flagellum, let me give you some of its operational parameters. Howard Berg, who works on the biophysics of flagellum, bacterial flagella, at Harvard, I've heard him speak several times at meetings and refers to the flagellum as the most efficient machine in the universe. And then he'll go through these parameters, that it self-assemblies and has repair mechanism. It's a water-cooled rotary engine. It's driven by proton motive force. It has 2 gears forward and reverse. Operates at 6,000 to 17,000 RPMs and can reverse direction within a quarter turn. So it's essentially a massless engine. We now have examples of bacteria that can spin their flagella at 100,000 RPMs. And this essentially can translate an organism at that speed, 50 micrometers per second, which is a phenomenal speed if you were to scale that up to our proportions. And this is pulling about 4,500 piconewtons per nanometer, which there's a lot of torque involved in the system. And it's hardwired to this signal transduction system with short-term memory, which I mentioned. Okay, so we're dealing with a true molecular machine. If we look at its composition, this is a microphotograph from David Desrosiers's lab at Brandeis, and this is the motor itself. We call this the hook basal body complex. The propeller would be attached to this structure here and extend on the outer surface of the cell. So this is out of the cell going into the cellular cytoplasm. LMP rings are referred to as bushings, like you would find in a true rotary engine. The hook is a universal joint transversing from the cytoplasm through the inner membrane and the outer membrane. Is a drive shaft that rotates. The engine is housed here in this C-ring, M-ring complex, and this would be acting as a stator. Okay, so we talk about the components of the flagellum analogously with true engines that we find that are made by man.
Bacterial flagella are composed of about 30 different proteins. First, a protein called Fly-F forms a rotor ring in the cell membrane. Fly-F self-assembles to form the ring, which then becomes the foundation for other proteins to attach, which in turn becomes the foundation for the attachment of others. The self-assembly takes place in methodical order through the accurate recognition of appropriate others. With a motor in place as the foundation, the filament that acts as the propeller is then formed of proteins called flagellin. With the help of a capping protein at the tip, flagellin molecules, sent out from the cell body through the central channel of the motor, stack up to form a helical tubular structure. The structure, revealed by electron microscopy, was very much like an artificial motor, with its stator, rotor, and bushing, and surprised the world. Image analysis of electron micrographs of straight flagellar filaments led to a three-dimensional image near atomic level. The central channel of the filament was extremely small, only 2 nanometers in diameter. Self-assembly of the flagellum, which grows out of the cell, always occurs at its distal growing end. The component proteins are produced inside of the cell and sent out to the tip through the central channel. The proteins are unfolded for insertion into the channel and then refolded at the distal end. Where then is the export apparatus? Research on flagellar protein export had been progressing extremely slowly until recently. All of these experiments have shown that flagellar protein export occurs in the following manner. First, fly I associates with two fly H molecules. A flagellar protein to be exported also binds to them, and this ternary protein complex binds to the C-ring and waits for the export gate to become disengaged. When the export gate is free and available, the ternary protein complex docks to the export gate, together with free fly HI complexes that are floating nearby or attaching to the C-ring. The gate is closed when it is free. However, it opens when the fly HI hexamer ring complex binds to it and efficiently inserts the amino terminus of the flagellar protein into the gate. Later, fly H and fly I detach from the export gate through ATP hydrolysis. The export gate utilizes proton motive force to send flagellar proteins into the channel. It unfolds flagellar proteins into long stretched chains for export into the 2 nanometer channel. Once the whole chain is within the channel, it is transported to the distal end of the flagellum. Meanwhile, the fly HI complexes that have detached from the export gate bind another flagellar protein and bring it to the waiting circle on the C-ring, repeating the cycle of binding and release for efficient export. The flagellar proteins exported in this way bind one after another to the distal end. The flagellum becomes longer, and it eventually becomes possible for the bacteria to swim. The rotation of the motor is transmitted to the flagellar filament with a gentle helical structure to generate propulsion. The hook acts as a universal joint, so that the torque can be transmitted regardless of the orientation of the flagellar filament. Bacteria can swim about freely because of this. What is the mechanism here? The structure of the hook was studied in detail to learn the secret. The hook is a tube of 55 nanometers in length, in which roughly 130 hook protein subunits are bonded together. This is the atomic model of the hook. The D2 domains of the hook protein are strongly bonded to one another on the hook surface to form the right-handed 6-stranded helices, much like a spring, and they form a kind of mesh structure with the D1 domains on the inside. This is what brings about the rigidity against torsion. The mechanism that determines hook length is as follows. First, hook protein molecules are efficiently exported, and the hook becomes nearly 55 nanometers in length. The autocatalytic cleavage of Flu-B slows down the export of hook protein. Then several fly K molecules are exported to measure hook length. In other words, the cleavage of Flu-B works as a molecular timer that controls the speed of hook protein export. As you can see, the precision mechanisms for constructing biological nanostructures are becoming clarified.
And of the thousands of papers written on this organelle, none address its mechanism of emergence or evolution. Okay. We don't have any intermediates. No one has postulated how this thing arose. And it's not out of ignorance. I mean, we know how this thing operates. And in fact, we have no mechanism to explain the evolution of such complex machines. So I would say intrinsic in this organelle is, design. And this isn't some God of the gaps type of argument. In any other situation where you see rotors and stators and U-joints and drive shafts, you would infer a designer. Okay, it's the simplest explanation. And I think it's going to be, the onus is now going to be upon Darwinian proponents to come up with the mechanisms whereby these things arose, because they are complex. Let me end with this. Okay, with as much as we know about this system, there's nobody yet that could build one of these systems with all the intelligence, combined intelligence. Okay, we still can't make molecular motors that can operate under these conditions of 100,000 RPMs or whatever. Right? So you're going to have to operate on a little bit of faith then if you're going to say that these things can be solely the product of chance and necessity. With that, I'll end it, and happy to entertain any questions. Thanks for your attention.